Dan Cortez-Perez is an associate research scientist in the Paralab at the Jackson Laboratory, and he is from Mexico. He is an MD and PhD from the National and Autonomous University of Mexico, which is UNAM by its acronym in Spanish. He came to the Jackson Laboratory as a postdoctoral associate, and I like to think that he loved it so much that he stayed on and is now a research scientist. I don't actually know if that's true, but I'm making up a backstory for Dan.
It is true.
Okay, it is true, good. And when I asked Dan what his favorite science movie is, this just made my day because he said it's Gattaca. And thank you. My husband and I are the only people I know of who've even heard of this movie and watched it, so it totally made my day. If you haven't seen it, I highly recommend it. It's a great movie. And his favorite science-based song is Lateralis?
Lateralis.
Lateralis. I swear I read this before. By Tool, which is based on the Fibonacci sequence. So Dan's talk title is How to Understand Our Brains Better Using Stem Cells and Mini Brains.
Can you hear me?
Yeah.
Yeah. Thanks so much for being here and for inviting me. My name says on the program that it's Cortex. That's not my name, it's Cortez, but I'm gonna take it because I actually work with the cerebral cortex. So I don't know why I didn't think of it. So first of all, I don't know if you know what a stem cell is, but basically a stem cell is a cell that can replicate itself many, many times. That doesn't happen in our body, that's why we die, right? So, but then the other property that it has is that it can turn into any cell of your body, any cell. You can make brain cells, you can make heart cells, bone cells, whatever you want. And you might wonder where they come from. So they come from an early embryo. We take the cells, we grow them on a dish, and that's it. Recently, there was this discovery. You can artificially generate these cells by taking cells from an individual. It doesn't matter if it's 100 years old. You go back in time 100 years and let them behave like an embryo, and they call iPS cells. And then you can differentiate them again into any cell of the body. So I think that's pretty cool. Actually, everybody thinks that that's pretty cool. So that's why the guy got a Nobel Prize. So, well, I'm not going to talk much about this because thanks, Marco. You introduced Ramon and Cajal. It was, I think it's our idol and our hero. Everything he did was with drawings. So how do you turn a stem cell into a neuron? So we basically follow developmental patterns. We're just cheating on what actually the body does. So we're just copycatting what the body does. So these are stem cells growing on a petri dish, and what you're going to see when in this preparation, anything that turns red or green is becoming a neuron. So this is over the span of 2 days, 5 days, 8 days, and if you can see, it's not just the change of color, but also the morphology. You can see the wires of a neuron there. And finally, this is how they look. So you can see these are human neurons going on a dish. And also, thank you, Marco, because many of the techniques you talk about, I also use them. We work both with the brain. So I'm not going to talk much about it. You're going to save me some time. So the other thing we do, and as he said, we are 3D individuals living in a 3D world. So by growing things on a 2D, you miss many of the talking of the cells and how they arrange and how it works. So growing organoids, it's a in vitro that's going to help us a lot understanding the brain. So just by changing, the simple way of saying this is that you can grow these cells in suspension in a bioreactor, and the cells, by giving the cells some cues, they will grow into a little brain. We call them cerebral organoids, but mostly, commonly they're known as mini brains. And this is one of the mini brains I grow in the lab. You can see that these colors represent the different types of neural cells that are in the brain, or also here in this mini brain. So why do I do this? Because I'm interested in stroke. Stroke is one of the leading causes of death. So basically, in case you don't know, it's basically the rupture of a vessel in the brain or the blockage. And what this is going to cause is the lack of oxygen and nutrients that is flowing into the cells. So how do you use these mini brains to study stroke? So what I do is like, this is an image of these neurons growing on a petri dish. And you can see that they're beautiful, they're interconnected, they have these beautiful wires, and then when I deprive them from oxygen and nutrients, they look like this, like all the axons are torn apart, they don't look as beautiful as they used to look. So by depriving them in vitro of those molecules, then you can mimic that stroke in vitro. So I found that some neurons were able to survive and others were not. So by doing analysis of the DNA and protein expression and RNA expression, I found that don't pay much attention into the details. I only want you to see that I found some genes that are important to the cellular response to hypoxia, which means basically just the lack of oxygen. So what I did is, what I was thinking is, how can we use these genes to, or activate these genes or gene pathways to make the cells survive after a stroke? So what and basically what you want to analyze is like the neurons, what neurons do is communicate. They send signals to each other via electrical impulses. So you can actually understand that in vitro. And by growing the cells on a sorry. I'm just going to let you know that we found some molecules that can survive. They can survive.
Thank you. Questions?
Yeah, could you just go through the rest of that slide and finish your thought? Thank you.
Okay, this is basically it. Each single square is a different treatment with molecules we're analyzing. And when you see signal, it means that the neurons survive. And where you see there's no signal, like in here, the neurons died. So we found some molecules that can actually make the neurons survive. And that's it.
When you were talking about the DNA analysis, you said you found some genes and gene expression that help the neurons survive the oxygen deprivation, but then here you were talking about recover neuronal activity. So do they need to have something activated like before you have the stroke, or even if they've already?
So what I didn't say is that I'm studying how genetic diversity affects or make the cells more resilient. So what I'm doing is using stem cells from different genetic backgrounds, from people from Asia, from Africa, from European ancestry, Latin American ancestry. So cells behave differently, and as like we are different. So like in my case, I'm brown because I express my skin cells express more melanin, and so on, so back and forth. So neurons are the same; they express different proteins in a different fashion. So I found that some neurons from certain backgrounds are more resilient to the stroke model. And now I'm trying to use those pathways from those cells to make the more susceptible neurons more resilient.
So you've talked about how you're using all these neurons and these grown cells for stroke behavior, and I'm wondering, have you ever thought about using them for anything else? Or
using what, sorry?
The neurons or the stem cells being grown. I'm wondering if you're using them, or even thought about using them for anything else.
Like what, such as?
Well, just
sorry, maybe I'm missing the.
No. You're using them to do research on strokes, correct?
Yes.
Yeah, I'm wondering if you've used them to do research on anything else. Or,
yeah. Like there's another thing I also study, autism. So but that's more a developmental issue. So what I'm analyzing is how there are many genes associated with autism. So what I'm trying to understand how those genes interact. And for that, I don't need the brain or the mini brains to fully develop. But I can I'm trying to do is use these mini brains to understand the different stages of development. So you can use them for many, many things. You can also, like, there's this cool paper that is totally unrelated, and it's like, they use stem cell-derived neurons, put them on a petri dish, connected to some electrodes, and they taught them to play Pong. You know that old? Yeah. So, and they learned in 5 minutes, and they get better and better. So,
wow, thank you.
Yeah.
So what do you foresee as the clinical applications? What are you wishing for clinical applications of the final results?
So in my case, is use these molecules to treat people suffering stroke. Right now, there's just one treatment, and that's it. And it's been the gold standard for 50 years. Well, less than that, 20 something. But there's nothing else. So there are many other things you can help with, but it's nothing that is going to cure these people. So we need to come with something. But stem cells by themselves, there's so many things you can do with them. Like another approach that we've been working with is use them as cell therapy in case let's say a heart attack, you meet that some of your cardiomyocytes die, and then you can transplant them. There are many things you can do with the stem cells.
How big is a mini brain?
I knew somebody was going to ask that. They're not that big. That's the thing. They're only like up to half a centimeter. So they're not that big. And one of the problems is that the bigger they grow, they need oxygen to get in. So they need the vessels, the blood to get in. Because usually how they get the nutrients and the oxygen is just by diffusion. And that's why we don't work by diffusion. We have blood cells and all that stuff. So that's the main problem right now. And there are many labs working on it.
Thank you.